Hello, my name is Christine Seidman, and I'm the Thomas W. Smith Professor of Medicine and Genetics at the Brigham and Women's Hospital. I also have the privilege of directing the Cardiovascular Genetics Center at Brigham and Women's Hospital. And today I want to tell you about a very important form of heart disease called cardiomyopathies. As you can see from these slides, when a heart develops a cardiomyopathy, it changes considerably from its normal structure. We generally characterize cardiomyopathies in two types of forms. One on the left called hypertrophic cardiomyopathy, in which the walls are thickened, and the other on the right, in which the walls actually appear thin because the chamber, the blood volume, is actually increased. These, as you can see from the statistics on the slide, are incredibly common disorders within the general population. We've known for a long time that when patients develop a cardiomyopathy, they have adverse outcomes. And these are important arrhythmias that can threaten their lives and cause sudden cardiac death. They also cause progressive deterioration of function, or what we call heart failure. As you can also see from this slide, the expenditures that we use every year in the United States to treat patients with cardiomyopathy are considerable. And unfortunately, despite this incredible investment by our economy, they often do not do anything to alleviate the progression of the disease. Indeed, cardiac transplantation is the only way of truly curing cardiomyopathies. And while these are life-saving interventions, unfortunately, only 1 in 10 patients with cardiomyopathy will ever receive the heart transplant they need. Moreover, in those fortunate enough to receive a transplant, their median survival is only 12 years after the transplant. So by no means are these a real cure. Now, for many years, we've recognized that there are genetic causes of both dilated and hypertrophic cardiomyopathy. And from lots of detailed investigations, we understand that there are 2 predominant consequences of these gene mutations. The first is haploinsufficiency, in which the amount of protein that is produced by the mutated gene is profoundly reduced, and so the normal level is 50% of normal. The other consequence is when the level of the protein is normal, but the function is substantially perturbed. And this abnormal function is actually what causes the disease. By knowing that there is a genetic substrate for any disorder, including cardiomyopathies, we really have a new and ultimate opportunity. And that is simply not only to predict disease development in those individuals who carry the mutation, but we have now the opportunity to prevent those diseases from ever developing. So how would we do this? Well, given the 2 different strategies that we understand cause cardiomyopathy, we need 2 different therapies to address them. For those mutations that cause a reduction in the amount of protein, we would want to supplement the gene and allow normal levels to be restored. For those mutations that perturb function, we want to silence the mutated gene so that only the normal gene copy will make protein, and hence, again, function will be perturbed. Well, that's very straightforward and clear, except for one major problem. Unfortunately, there are many challenges to correcting these gene therapy approaches for the following reasons. The first is that many cardiomyopathy genes are simply too big to directly deliver to the cells that need them. For example, titin is a gigantic protein, encodes 35,000 amino acids, and the gene that encompasses it is more than 38,000 nucleotides. We can actually see this protein under the light microscope looking at cardiomyocyte striations in which 2 titin molecules span those different striations. Moreover, titin truncating mutations that result in a 50% reduction accounts for about 20% of dilated cardiomyopathy, a substantial contributor to this disorder. So what are we going to do instead? Why, we're going to activate the other endogenous gene that is not mutated. We can now use molecular strategies to harness the transcriptional promoter and increase the expression of the non-mutated gene and restore normal protein levels. Well, what about those mutations that cause dominant negative, or they reduce the function without perturbing levels? An example of that is the myosin heavy chain gene, in which there are literally hundreds of unique particular mutations that are specific to a family or a few individuals. And these account for almost half of all hypertrophic cardiomyopathy cases with a genetic cause. So what we'd like to do now is to not correct each and every one of these individual mutations, but rather to silence a common variant that is present on many people on the same gene that carries the mutation. And that's illustrated here. I'm showing you in green and gold 2 different mutations. If we inactivate this gene, as indicated by the X, we will get rid of all of the mutations that are downstream of that inactivation signal. We've actually done this now in mouse models. And as I hope you can appreciate, on the right is what happens when the gene is silenced, when it has a normal myocardium without any accumulation and fibrosis, in comparison to the untreated animal in which that blue material in the heart section and the disordered array clearly shows evidence of disease. Well, there are many new opportunities going forward, but there are also going to be many challenges that we have to address. We need to be able to establish the best therapeutic strategies, and we will do that using induced pluripotential stem cells and large animal models, as well as our mouse models. We need to be able to demonstrate functional efficacy, and certainly the appropriate methods for delivering these therapies to our animal models, as well as our patients. Equally important, we need to be able to understand if there are any off-target, unintended manipulations of the genome when we deliver these therapies. And most importantly, we need to figure out how to do this not only in experimental systems, but safely and effectively in humans. Here at the Brigham and Women's Hospital, we have the opportunity to do that through the Cardiovascular Center and the investment that we've made in understanding this disease, the mechanisms by which mutations cause disease, and the patients and other researchers who are committed to solving this very important problem. Thank you for your attention.